Hello, good day, and welcome again to the channel. Today we continue in our tutorial series, and I'll yet show you another important feature in PyMOL. Specifically, this is important if you have maybe a comparative analysis of several orthologs of a particular enzyme. The one common challenge you will face is the fact that the residue numbering in these orthologs, even if they are the same enzyme but from different species, may not be the same, especially when the crystallization is done by different laboratories. For this tutorial video, I'll be using the acetylcholine esterases, a key therapeutic enzyme in Alzheimer's disease and also an important molecular target for insecticides. So for this study, I have 4EY7, that is the PDB ID. You can see I have already uploaded them here. 4EY7, that's from Homo sapiens. 6ARY, Anopheles gambiae. 6G1U, T. californica, and 6SYU from Drosophila melanogaster. Catalytic efficiency is critical in this enzyme, and for this reason, it has a highly conserved three-dimensional structure. So the enzyme has a deep, narrow, active site gauge. The active site, active site gauge, comprises two main components: the catalytic active site and the peripheral anionic site. So if you're interested, like in this my case now, I want to look at molecular interactions between these enzymes and a specific ligand or several ligands for a comparative study. You will see that I will need to understand a whole lot about these enzymes so that the definition can be scientifically presented. So the first thing you want to do is to first superimpose to see if these enzymes structurally are the same. And then once that is established, you now check for the matching amino acids. So stay with me as I take you through this one. For us to start, let's now first display the sequences. So you can see all the sequences: 6ARY, 6G1U, 6SYU, 4EY7, and then the numbering. So like I said just now, you can see what's happening here. The first one here for chain A is 163. The first one here is 4. This is 3, and this is 4. And even at that, you can see that the amino acids are different. So to make it easy, let's use the human enzyme 4EY7 as our reference enzyme, and then let's superimpose and first confirm how the RMSD, just to check structural compatibility. So I'll just say align in this command phase, align 4EY7 underscore A. Okay, after cleaning, I saved it here as A just to indicate that it is this thing. Align 4EY7. Let's start with 6ARY. 6ARY. Enter. Okay, you can see the value here, 0.613 executive RMSD. So it means structurally you don't have a problem with that. We'll go for the second one. I'll just call this one up and then say 6G1U, G1 Okay, now we have also 0.645. So you can see that all the ROMSDs are less than 1, 0.902, and then atoms matched there. You check for the other one, the same thing, 0.645, and then we have 0.613. So once that's done, we now want to confirm specifically the matching amino acids. To check the matching amino acids, since we already started using the 4EY7 human enzyme as our reference structure, we will first identify the amino acids of interest. In the catalytic triad, that is in the catalytic active site I gave us an example earlier in this video, comprises serine 203, histidine 447, and glue 334. So I will identify those residues in 4E17 and compare them with the other ones. But first, what I will do is hide these two so that it is easy for me to identify the particular one of interest. So in 4EY7, that is this second one, I will just go to serine 203. Okay, this is 201, 202. So you can see that as I click this one, look at this base. You can see this is the whole matching sequence, irrespective of the length you have here. The whole sequence is in this lower bar represented by symbol bars. If I click this, for example, and this, you will see that the two of them, they are here. Yes, you can select, and this is useful during cleaning. You see, anyone you select becomes highlighted. And because I'm comparing two, you see that the space you have is just for half of these two. Okay, but that's not what we want to do. We will now deselect. Okay, we need that one. Let's come back to the other ones here. Please deselect. Let's drag your mouse over. Click and drag. So now we have serine 203. It's here. And then you want to check what is. You can see the numbering for 6AROY is very different. So what is the matching amino acid here? Most times the PyMOL already helps you align it. Remember, we first had to confirm structural compatibility. So if our ROMSD was not good at all in terms of the value up to 2 and above, which is indicating that there is a strong structural difference, personally, I do not advise this. So our interest in this particular study, for example, is to see, okay, how the specific amino acids, first, the amino acids that are present in the other enzyme in the same position. Then, if I have selected serine 203 here, if I click this, for example, just watch out, you will see that the green residue is hanging somewhere else. So it means that is not a matching amino acid. So take this one off, then just come on top of it. Then watch out, you see. So in the two structures, where you have serine 203, it means you have glycine 362. So you can take note of that. And then by the time you map the particular amino acid, when you draw your grid coordinate around it, now check to see. So it's just something you do out of understanding, and it will be very useful in explaining the molecular interactions between the ligand and the enzyme, especially if you're using the same ligand for both structures. Once you draw the grid, you see that the matching amino acids, like I've been emphasizing, coming from the fact that you have the same enzyme structure. So the next is okay, let's take glue 344. So we can come down to glue 344. 340, okay, 41, 42, 43. Is that it? 334, sorry. 331, 332, 333, 334. So the matching here will be, so you can already check. No, it's not. We didn't get that one this time. Take this one off. Why? That should be tyrosine. Yeah, so it's matching. So you just take your time through, since you already know the residues in your reference enzyme, to confirm which residues are in the other structure in that same position. You just go through all of it like this. So in this case, for example, I'm interested in the catalytic triad, which has its own residues, the Oxy anion hole, and the AC binding pocket. These are different subsites within the active site gauge. And then we also have the peripheral anionic site. So since the reference enzyme already has all of these amino acid residues listed out, I can now just trace in the other one. But like I said, you have to now go further by trying to view the binding pocket, draw a grid around it, and then confirm to ensure that the amino acids that you have selected are actually in that pocket. One easy way I identify the interacting amino acids in or between the enzyme structure and the co-crystallized ligand, in this case for 4EY7, I said that's the reference structure, is to come to this discoveries to the visualizer, and once you select the ligand group, as you can see here, so what I have is just the 4E7A with the co-crystallized ligand, but I've already cleaned it completely here. So once you select this, I'll just say from PDB site records, okay, if that won't even work, select ligand group and current selection. This is too big now. Let's delete that. Okay, also from receptor cavities, we have several receptors now. So let's confirm if, or several cavities, if this is covering, yeah, it is covering it. So that's site 1. You can go to site 2, no, site 3, no, site 4, no, more, just on and on. So site 1 is good for us. Then what we'll now do is, let's see the SVD spare. Then quietly come here and show and hide. So once this is done, come to A, and then you want to check the amino acids within this sphere. So our interest first is serine 203. So let's go there. Let's see if serine 203 is there. Perfect. That is it. That's serine 203. Yeah, next is glue 334, and we can see it here. And next is histidine 447. So you can see that the example I just picked now for the catalytic triad, that is serine 203, histidine 447, and glue 334, they are within this sphere. So we can go ahead now and copy this information. For the sake of this video, I'm using just one pocket. You can go through, I mentioned 3 sub-pockets under the catalytic active site and the peripheral anionic sites. You can just, based on information from your literature review, you can just go ahead and check all the different sub-sites to ensure that it is captured within, or the amino acids are captured within this your sphere. So once that's done, you can come here, just select this, you have this sphere, right click, and then attributes of the site. So you have your coordinates here, XYZ coordinates. So since you have previously superimposed and found that the enzymes are structurally compatible, you can now use this to build a grid on all the enzymes to conduct your docking. That way you are able to check, that's restrict the docking to that particular site, and can compare across the different enzymes.